All right, hello everybody. Hope everybody is staying safe. We just got our order to stay at home. That's really frustrating. And it's okay to be frustrated. It is totally okay to be frustrated. In fact, you should be frustrated because, it's a hard thing to do to balance, what's good for a lot of people versus what it is you want to do. And that's like a real emotion. Acknowledge it, but then remember that we're doing this to prevent something huge, to prevent something gigantic happening, and to protect some of those that are most vulnerable. So I'm happy to still be seeing and talking to all of you through all kinds of different ways. And today will be a little bit of a break as far as notes go, because most of these notes are review and just sort of clarifying some things about DNA replication and meiosis. The very end will be new things. Did I say just now? Okay, most of these notes will be like a review, not new. And the very end will be kind of some new stuff that most of you are already familiar with, but just a few new things. Okay? All right, so let's start off by reviewing some DNA replication things. Okay. Let's see here. Okay. So first I'm going to zoom in on this excellent picture showing DNA replication. There's two enzymes that are associated with DNA replication are helicase and DNA polymerase. Remember that DNA replication happens semi-conservatively, which means that each new DNA molecule will be half original DNA and half new DNA. And remember, we've got our template strand and our synthesized strand there. So first of all, the helicase. The helicase is what unwinds the DNA. It breaks the hydrogen bonds that are holding together the A's and T's and G's and C's and kind of unzips the strand and opens that strand up. So now that we have these open DNA bases, we need a molecule that will come along and put together the complementary base pairs from these nucleotides that are just floating in the nucleus here. So the DNA polymerase just kind of zooms along this original template DNA strand and grabs free nucleotides from its surroundings, puts them together with their complementary base, and then covalently bonds the backbones together. There's a couple DNA polymerase molecules happening at the same time. One zooms this way and the other one zooms this way. Next year you'll learn why they go in opposite directions. But that's just the point of these molecules. So this is an enzyme because it ends in ase, and it's building a big new molecule which is called a polymer, and so that's why it's called polymerase. Remember that Meselson and Stahl did a great experiment, one of the most famous experiments in all of biology, that showed how DNA replicates. Is it conservative? Was their first guess. Is it semi-conservative? Their second one. And is it dispersive? So in this case, the blue represents a bunch of DNA that contains nitrogen-15. So they grew a bunch of bacteria in agar that just contained nitrogen-15 until eventually all of the DNA was made of all of its normal atoms: carbon, hydrogen, oxygen, nitrogen, phosphorus. But every single bit of nitrogen in there, in those bases, in those A's, T's, G's, and C's, every single bit of nitrogen was made of nitrogen-15. And then they took that same bacteria and put it inside of some agar that had only nitrogen-14 in it. So as the bacteria would feed on this agar and replicate, because the bacteria would then reproduce themselves, so they'd have to replicate their DNA. As they did that, the food that they ate only contained nitrogen-14, and as a result, all of the new DNA that was made was nitrogen-14. So this yellow DNA is DNA that has all of its normal elements, carbon, hydrogen, oxygen, nitrogen, phosphorus, but all of the nitrogen in this case was nitrogen-14. And they did this so that when they spun it in a test tube and exposed it to some camera film, they could see layers in the test tubes for where these things were. So in conservative replication, after one round of replication, if DNA represented conservatively, and remember, these are all ifs right here, the half of the DNA would be nitrogen-15 and the other half would be nitrogen-14. And after a second round, that original strand would stay together, but all of these new strands that were made were still made from the from that agar that the bacteria were eating containing nitrogen-14, which means that we should see more and more bacteria being made all of nitrogen-14 DNA, and some of the bacteria would still retain that original set of DNA that had nitrogen-15 in it. And this is a big if. If DNA replicated conservatively, that's what we would see. And it would look a little bit like this. If DNA replicated conservatively, we should see this nitrogen-14 band at the top and this nitrogen-15 band down here. And after a second round, we would still have a nitrogen-15 band because that original parent strand would stay together. But we should see a bigger and bigger and bigger nitrogen-14 band. If DNA replicated semi-conservatively, that means that this strand right here would unzip, just like it would over here. It would unzip, and each new DNA strand would be half nitrogen-15, half nitrogen-14. But after 2 rounds, this DNA strand only has one nitrogen-15, and so the new DNA strand that's made would be nitrogen-14. So this, just to be clear, this is not conservative. Rather, this DNA strand right here is this one here, and this yellow DNA strand is a brand new synthesized DNA strand, but nitrogen-14 is the only one available. This nitrogen-14 strand is this one right here, and this, we follow the right one, yep, and this nitrogen-14 strand on the other side is new synthesized DNA. So, if that's what they saw after one round of replication, they would have seen one halfway in between the nitrogen-14 and nitrogen-15, because this one has a mass that's a lot lighter. And then after another round of DNA replication, our new DNA strands that are made from that nitrogen-14 template, that new DNA that's synthesized is also nitrogen-14. So we still see one halfway through, and then we see one just in the 14 level. That dispersive one is pretty crazy, and it's hard to kind of label everything, but it's easy to see. And then Meselson and Stahl's actual results lined up perfectly with semi-conservative replication. So these guys made a bunch of predictions about what they should see if different models happen. Of course, it's entirely possible that the results that came out didn't match any of their predictions, in which case they would have to say, well, none of these ideas are how DNA replicates, and so we're going to have to come up with some new things. How does this happen? And it just so happens that they made a very, very, very good educated guess on what the different possibilities were. Okay. That's some review about DNA replication. Remember, this happens in the S phase during interphase, no matter whether you're doing mitosis or meiosis. Okay, let's do our next round of review by drawing a whole set of all this different sets of stages of meiosis. Let's do a real simple cell this time. Instead of an N of 3, it's just going to have an N of 2. Remember that N refers to the number of unique chromosomes, so we're going to draw one short and one long. One short and one long. And then you're going to need a couple colors. Ready to go. Crayons, markers, pens, or light pencil and dark pencil, however you need to do it. Because remember that your cells that your sperm and your eggs come from start with 2 of each DNA strand, because you've got 2 of each DNA molecule, because you've got one maternal set of DNA and one paternal set of DNA. So start by just drawing the cell in G1 and G2. I'm going to pause my video as I'm doing this so that you can pause yours. I don't know what that means. That was weird. I'm gonna pause my video and draw G1 and G2 so you don't have to watch me draw it. But right now you should pause it and go ahead and draw those 2 cells and then check back. All right. So I've got my original cell right here. I've got one short blue, short red, one long blue, long red. And then after S phase happens, I have 2 short red, 2 short blue, 2 long red, and 2 long blue. Forgot to label this one as my G2. Let's assume our cell checkpoints have happened. And everybody go ahead and draw prophase 1 and metaphase 1. And in prophase 1, I want you to show the genetic recombination after crossing over has happened on the long chromosome. And then in metaphase 1, I want you to draw the chromosomes so that each daughter cell that is formed will have different maternal and paternal combinations. All right, go ahead and pause your video. All right, so here we go. I've got my nucleus breaking apart right here, and the membrane is starting to break apart. I've drawn my centrioles starting to make their spindle fibers. I've paired up my homologous chromosomes. So the blues and reds are right next to each other. I've shown my crossing over right here. And that's my prophase 1. In metaphase 1, they should be lined up and along the middle. And I've got my chromosomes in their tetrads right here. And I've shown my recombination right here by making this red chromosome have a little chunk of blue on it, and this blue chromosome having a little chunk of red on it. But I'm going to put a little star here. I'm going to put a little star right here, and I'm going to zoom over here to where I have a little bit of free space. Remember that there's that thing called random assortment. Random assortment, and that is that while we've got our cells here, I'm going to skip the recombination for just time's purposes right now. This is one possible way, and that's what I drew down here. This is one possible way that this metaphase could happen, and if this is the case, then this side over here is going to have short blue, long red, and this side over here is going to get short red, long blue. But remember our formula. 2 to the n will tell us how many different possible combinations there are in this. And that'll impact like what happens as far as which genes each sperm and egg get. And since in this chromosome our n is 2, we would have 2 squared in this case, which is 4. So there's 4 possible combinations. So pause your video, take a second, and try to sketch out what the 4 possible combinations are. All right, let's see how you did. Now, this is going to seem a little weird, but these could just be flipped so that the red is on the left and the blue is on the right. And this flipped so the red is on the right and the blue is on the left, which is how we get this one right here. This is a possible orientation of these chromosomes. We could have them so that the blues are on the left and the reds are on the right, or so that the reds are on the right and the blues are on the left. Either way, you end up with the chromosomes going to different sides and creating new possible combinations. So those are the different ways that random orientation could be drawn. How do you need to draw yours? Doesn't matter. One of those four. That's it. Oh my God. Okay. Everybody go ahead and pause your video and finish this whole meiosis up. Go ahead, pause it, and come back when you think you've got it. All right. So I've finished mine up. It took me about 10 minutes or so. So if it took you about as much time, then you're right on track. But go ahead and pause the video right here. Check yourself all the way through anaphase 1, telophase 1, cytokinesis 1, prophase 2, metaphase 2. Go ahead and pause your video. Check yourself. All right. I'm going to scroll over now so that you can see anaphase 2, telophase 2 and cytokinesis 2. Go ahead and pause your video and check yourself. Okay. So the result is over here that we have 4 haploid gametes. And gametes are another way to say sperm cells, egg cells. Sometimes, and this is a little old school, but sometimes these are referred to as germ cells, which is really, really weird and really old school. But when you hear that word germ, what that's referring to is where things come from. And since human beings arise from, these combinations of sperm and egg and so forth, then that's kind of why they call them that. As to when that germs became things like bacteria and viruses and stuff that cause diseases, I don't know. Maybe it's from when you get a disease from something else, and so the thing you get the disease from is like where it started, and so that's why they're called germs. Who knows? If only we had science. Okay, now here comes some of the little bit of a new thing. Okay, I should make a note right now that all of the new stuff starts at about the 18-minute mark. Okay, so I'm going to go way back here to cell number 1. This is our starting cell. This is the one that all of the sperm and eggs are going to be produced from, okay? So this is cell number 1. Now, let's imagine for a second, and let's just focus on the little blue chromosome, okay? And let's imagine that... Go away. Okay, let's imagine that right there was a gene. And that gene was a code for, we'll say, blue eyes. Okay? Now, because the red chromosome is homologous to that blue one, then at the same spot, there's also going to be a gene for eye color. Now, let's say that this one was a gene for brown eyes. Well, it just so happens that in human genetics, brown eyes is what is called a dominant allele, and blue eyes are governed by a recessive allele. And so we give little codes for these things. And just to make things easier, we always make these things a single letter. And we always use a capital letter to represent the dominant allele and a lowercase letter to represent the recessive allele. So if here on this blue chromosome we have a gene for blue eyes, we're going to label that as being a little b. And then right here on this red chromosome, if the allele is for brown eyes, we're going to label that as big B. Now, that means that this individual, as far as eye color, has a big B allele and a little b allele. Now, these words, recessive, dominant, and allele, are from the reading that I sent out yesterday. So it's important that you have read that and you understand these words that I'm talking about. Okay, so as we go all the way through meiosis, and we get all the way down here, that means that if this sperm or egg is passed on to a child, it is going to receive that little b or blue-eyed allele. The same with this one here. But if this sperm or egg gets passed on to a child, that means that it will get the brown-eyed or big B allele. And the same is right here. So let's imagine for a second. Now, that means that we've only got two possibilities. Our kid is either going to get a dominant brown-eyed allele or a recessive blue-eyed allele. So we have two possibilities. So let's draw a little sperm here. And let's imagine that this sperm is carrying the recessive blue-eyed allele. And let's imagine that this sperm right here is carrying the dominant brown-eyed allele. Well, as you know, you can't choose which sperm you pass on, and you can't choose which egg you pass on. That would be pretty cool if we could, but that would probably be really expensive, disproportionately affect the rich, and once again, poor people are left in the dust, and we don't want that to happen. So let's just imagine that we still live in this world where we can't do this. Now, let's imagine that this big B, little b individual mates and has a child with a big B, little b individual as well. You know, of course, they've been together for a while. They make a nice, mature decision. You know, they're ready to realize that kid should be number one, and you know all that kind of stuff. But all right, so let's put this egg right here. Egg cells are always a lot bigger than sperm cells. And let's make this egg cell right here. So that same individual, when they go through all of meiosis, just like this right here, when they go through all of meiosis and they make all their sperm and egg cells, when they make all their egg cells, half of their egg cells will have the little b allele, and half of their egg cells will have the big B allele. So what happens if this sperm fertilizes this egg? You end up with a zygote that is big B and little b. Now remember your readings from the other day. And that means, and that is saying that if there's a dominant allele and a recessive allele there, the only gene that gets expressed is the dominant allele. This would be a brown-eyed baby. Now, they're still carrying that recessive allele, and they could pass that recessive allele on to their children, but they're brown-eyed. Now, let's imagine that this little b sperm fertilizes this little b egg. We end up with a little b child. What color eyes does this child have? That is a blue-eyed child, because in order to express the recessive allele, you must have both recessive alleles. Okay? Let's imagine this big B sperm fertilizes this big B egg. We end up with an egg cell that is big B, big B. Now, this individual isn't super brown-eyed. This individual is just brown-eyed, but it just so happens that they carry both dominant alleles. And then finally, if this big B sperm fertilizes this little bee egg, we get a zygote that is big B, little b. This is also a brown-eyed child. So, what are the chances that this pairing produces a blue-eyed child? And that's a 25% chance. Now, statistics doesn't mean that you're going to have, if you have 4 babies, that one of them will be blue-eyed. It's totally possible that all 4 could be blue-eyed. But that's because each time this couple has a child, this probability completely resets. Every time they have a child, this probability resets. So there's no, well, I already had a blue-eyed child, so it's got to be brown-eyed the next time. I mean, think about it like a coin flip. If you flip a tails one time, does that mean you have to flip a heads the next time because it's 50-50? No. The two things are completely independent of one another. Now, you read about Gregor Mendel, and Gregor Mendel was the first one to figure out that there's patterns to inheritance. And that is a really important note to make right now. There are patterns to inheritance. And that means that we can kind of predict some of the things that are going to happen as far as inheriting characteristics from your child. I'm sorry, from your parent. Ooh, that's weird. And Gregor Mendel figured this all out without knowing what DNA was. If you read yesterday's information, you read about his experiments with peas. He grew peas, and he figured out that, like, when you combined plants that were tall with plants that were short, you always got tall plants. But if you bred those plants from that original grouping with each other, some short plants all of a sudden came out. And that's what he was figuring out. He was, in fact, breeding big B, little b plants with big B, little b plants. Now, these plants would seem to be all tall, but just like this right here, there was a chance that the short plants would come out. So he was showing that genetics can follow some patterns, and therefore, in some degrees, it is a little predictable. So, we have terms for some of these particular combinations. And let's define a couple of words right now. When you have two of the exact same allele, like this one right here, or like this one right here, you are considered to be what is called homozygous, which just means that you have the same alleles. And so this could be big B, it could be little b, I mean, it could be any letter. So, if you have different alleles, I bet you can predict what this word is. You are what we call heterozygous. And in this case, the only option for that is big B, little b. Now, some of you may be looking at this picture right here that I just made, and you're thinking, that looks like something I've seen before. And maybe you've seen this picture written in a slightly different way. And instead of drawing the sperm and the eggs, you are making a little chart for yourself where you put the parent alleles right here and right here. The top represents the two possibilities for the parent one allele, and the side represents two possibilities for parent two alleles. And down here in these boxes are the possible children that could be the result of this cross of parents. And you may even know the name of this picture that's right here as it being called a Punnett square. Now, FYI, IB calls this a Punnett grid. Same deal. But a Punnett square or a Punnett grid is a way to take the parent genes and pair them up with each other to see what the possible offspring are. And that's what all of these guys are right here. These are the possible offspring. Okay, so that's it for today. Aside from this review here, I've got one reading for you and a little practice worksheet with an answer key. Everybody stay safe. Everybody try to explore something new. Amazon has sent out free online classes for how to learn how to computer program. I think that's pretty cool. There's something for middle schoolers and something for high schoolers. I think my son is in 5th grade. I think I'm going to open up the middle school one and maybe try to do it with him. I taught myself how to make websites once, so I think I can do this. All right, everybody take it easy.